cloned sheep, cloned dogs, clone wars. In 1903, Herbert John Weber, perhaps the most notable plant breeder and botanist in the United States, coined the term clone to refer to the technique of propagating new plants using cuttings or bulbs. Later, science fiction writers like Huxley, Toffler, and Levin used the term clone to describe making replicas of humans. Let's clone something today. Hi, I'm Lenny Rome, the Vault Guy. I'm a cell biologist and a nanotechnologist. My passion is a common particle of the human cell called a vault. My goal is to explain what vaults are to non-scientists. This is episode 7 of the Vault story. If you're new to my channel, you might want to start at the beginning where I describe the nanoscale. If you haven't seen it, or the other 5 videos, I'll put links below. Today, I want to cover 3 things. First, I want to remind you what genes are and review the process by which a cell reads a gene so it can make a protein. Second, I'll explain why we set out to clone the gene for the major vault protein and how we achieved our goal. Finally, I'll tell you what we found when we compared the major vault protein to every known protein that had ever been identified. Stick around and see if I can actually help you decipher the genetic code. I think I can. Once we had determined that the vault particle was made from 3 proteins and a small RNA, my postdoc Valerie and a new graduate student Sanjay Vasu both focused their attention on the 100 kilodalton protein that we called the major vault protein, MVP. Sanjay went after MVP in the primitive organism slime mold. I'm going to save that story for a later video. Val set out to clone the MVP gene in a mammal. In order to explain to you how we went about characterizing the vault proteins, I need to review your knowledge of molecular biology, especially how genes relate to proteins. Don't worry, this won't be on the final exam. Let's start with the basics of molecular biology, which I feel really got started in 1953 when Francis Crick, James Watson, Rosalind Franklin, and their colleagues working in Cambridge, England, created the double helix model of DNA. Here's a famous photo from that time. Crick is on the right. By the end of the 1960s, less than 8 years after the Watson-Crick model, the genetic code had been broken, and we had a basic understanding of protein synthesis. We now know that a gene is the basic unit of heredity, and that genes are the instructions for building each protein of the cell. So if you forgot this stuff when your high school biology teacher taught it to you, or if you never really understood the process, allow me to give it a shot. In my second video, I made the analogy that the cell nucleus is like a central library. Each of the books in this library are called genes, and each gene is the instructions for making one of the proteins of the cell. The instructions are written in DNA. Unlike an actual library, the books in the nucleus are attached to one another, and they don't leave. Instead, when the cell wants to make a particular protein, it makes a copy of that specific book of instructions, and the copy leaves the library, goes into the cell cytoplasm, where the copy of the instruction book is read and the protein is assembled. Let's look more closely at the process of making a protein. As I just said, it begins in the nucleus, where the instructions are stored in DNA. DNA is made in long double helical chains from building blocks called nucleotides. Nucleotides are molecules made from a sugar phosphate backbone attached to a nitrogen-containing base. The sugar is deoxyribose, and there are 4 different nucleotide bases used to make DNA: adenine, thymine, guanine, and cytosine. Think of them as the 4 letters of a language. The letters are A, T, G, and C. abbreviations for the 4 nucleotide bases. Each of the nucleotides can bind to one other nucleotide. A can bind to T, T to A. C can bind to G, G to C. Think of it like A and T love each other, and G and C love each other even more. When you look at a DNA chain, you see that the sugar phosphates are attached to one another, forming a backbone, shown in this cartoon in purple. There's an opposing strand of DNA that's running in the opposite direction, and the 2 strands are held together by these ATGC attractions. In DNA, these strands are twisted around each other, forming a double helix. In the famous photo I showed you, Watson and Crick have built a stick model of DNA. This model was based on Rosalind Franklin's data, but that's another story. In this photo, Crick is pointing to the double helix backbone, held together by the ATGC attractions. You can see on the wall behind Crick is a drawing of the DNA double helix in a style like the cartoon I drew. What a cool Easter egg. So let's say you want to make a particular protein, like MVP. You first need to make a copy of the gene for that protein. Let me illustrate this by returning to the cartoon of the DNA double helix. Let's unwind the helix to make it easier to see what is happening. The first step is to make a copy of the DNA into a form that is mobile and can leave the nucleus. This process is called transcription. A simplified version of the process is that the 2 DNA strands around the gene are separated and a copy of DNA is transcribed from a similar material called RNA. The RNA backbone is a little different, so I colored it yellow here. The copy is called messenger RNA, mRNA for short. Like DNA, RNA is built from a nucleotide backbone. However, the sugar is ribose instead of deoxyribose. 3 of the 4 bases, A, C, and G, are the same as in DNA. Thymidine, the T, however, is replaced by uracil, U, in RNA. One RNA contains the step-by-step instructions for making one protein. The mRNA then exits the nucleus and enters the cytoplasm, where the instructions are read and a protein is produced. This process is called translation. As I explained in episode 6, proteins are built up from basic assembly units called amino acids. The amino acids are attached together in linear arrays, like beads on a string. There are 20 different varieties of beads, amino acids. How does the cell know the sequence of amino acids that need to be assembled? This information is contained in the mRNA in the form of 3-letter words that are spelled out by just 4 different letters, the 4 bases of RNA. So basically you have a language that just has 3-letter words and only 4 different letters to work with. So naturally, you might ask, how many different 3-letter words are possible in a 4-letter vocabulary? The answer is 4 to the 3rd power, or 4 times 4 times 4, which equals 64 different words. Here's a table with all the possibilities and the translation of the words into the various amino acids. The 3-letter words are the codes for the different amino acids. And some of the amino acids are encoded by as many as 6 different words. One of the words, AUG, is called a start codon, which starts all proteins with the amino acid methionine. 3 of the words, UAA, UAG, and UGA, are called stop codons. These codons signal the translation to stop. So, how does translation work? Translation is carried out in the cytoplasm by a machine that we've talked about before called a ribosome. The ribosome moves along the mRNA and reads the 3-letter words sequentially. As the ribosome moves along, it couples amino acids together in the sequence designated by the order of the words. This is an incredible process, and there are several great YouTube videos that animate the movement of the ribosome along the mRNA, and the generation of the new growing chain of amino acids as the protein is produced. I'd like to make one for you, but I don't have the budget for a fancy animation. And decent ones are easy to find. Before the relationship between genes and proteins was understood, biochemists interested in a protein had to study the protein directly. What I mean is they would have to isolate enough of the protein from an organism and measure the protein's properties. Unfortunately, some proteins are very sparse, and their purification can be quite challenging. Shortly after the molecular biology revolution, biochemists realized that by cloning the gene for a protein, we could read the sequence of the nucleotide bases, and knowing the genetic code, we could decipher the amino acid sequence of the protein. So, with this basic understanding of the relationship between genes and DNA and proteins, let's return to the SDS page gel of the vault components that we described in the last video. The gel told us that the vault particle was made up from 3 proteins and a small RNA. So of course we wondered if any of these components had been described before. As I mentioned when I started today, Valerie decided to go after the gene that encodes MVP. Since we had an antibody that my former postdoc Nancy had made against vaults, and that antibody turned out to be specifically directed at MVP, Val used the antibody with a common gene cloning method called expression cloning. By cloning the gene for MVP, we could determine the gene sequence and thus infer from the genetic code what the protein sequence was as well. Scientists are, for the most part, very open and collaborative, so databases have been set up where all gene and protein sequences published, and sometimes even unpublished, are deposited and curated. Thus, we could search all the available gene and protein databases for proteins related to MVP. And to our surprise, or maybe I should say annoyance, we discovered the major vault protein was unique. Not only were there no matches to previously cloned genes, but there were also no related genes or proteins in any of the available databases. Thus, we had no clue as to what the MVP protein might be doing. I'm Lenny Rome, the Vault Guy. Join me in the next episode where I describe how we used the MVP gene to go fishing for the other vault proteins. Finally, do me a favor. If you find these videos useful or interesting and you'd like to see more of the Vault story, subscribe and give us a like. As my Nana used to say, it won't kill you to show a little appreciation.